Most people assume they already know this comparison. Black Americans descended from West Africans. Strip out the European DNA that slavery forced in, and what's left should look essentially identical to the genome of someone still living in Nigeria, Ghana, Sierra Leone, or the Congo today. The data says otherwise. When geneticists compared Black American genomes to West African genomes, marker by marker, haplogroup by haplogroup, immune gene by immune gene, the relationship was simultaneously more specific and more different than anyone expected. The West African ancestry in Black American DNA doesn't just point to a continent, it points to exact ethnic groups. The Yoruba and Igbo of Nigeria, the Congo of Central Africa, the Mende of Sierra Leone, name peoples traceable in the genome with precision that slavery tried to make impossible. 400 years of the Middle Passage, plantation mortality, forced reproduction, and a completely foreign disease environment left measurable marks on the Black American genome that distinguish it from its West African source populations in ways science is only now beginning to fully map. One gene alone, a POL1, tells the entire story. Evolved in West Africa over thousands of years to protect against sleeping sickness, carried across the Atlantic. Now, in the absence of the parasite, it evolved to fight, contributing to kidney disease rates in Black Americans that are 7 to 10 times higher than in European Americans. Drop a comment where you're watching from and hit subscribe. Let's start with the baseline, what the data shows about West African genetic diversity itself. Because before you can understand what happened to Black American DNA, you need to understand what Black American DNA came from. And the answer is staggering in its complexity and depth. Africa is the most genetically diverse continent on Earth. This is not a contested finding. Every major genetics institution, from Stanford to the Max Planck Institute for Evolutionary Anthropology, confirms it. The genetic variation within African populations is greater than the variation between any two non-African groups anywhere in the world. The reason is ancient and simple. Modern humans evolved in Africa and spent the vast majority of their 300,000-year history there before any population movement to other continents occurred. Every non-African population on Earth, every European, every East Asian, every indigenous American descended from a small group of migrants who left Africa roughly 60,000 to 70,000 years ago. They left with a genetic subset, a single chapter, copied out and carried away. Africa kept the full library. West Africa in particular is a region of extraordinary genetic depth and complexity. Over 2,000 ethno-linguistic groups live on the African continent, and a significant proportion of that variation is concentrated in the West and West Central African region from which the enslaved ancestors of Black Americans were primarily taken. The Yoruba of southwestern Nigeria, one of the largest ethnic groups on the continent with a civilization history stretching back thousands of years. The Igbo of the Niger Delta, whose decentralized society and distinct cultural identity made them one of the most heavily targeted groups in the trade. The Mandinka and Wolof of Senegambia, master rice cultivators whose agricultural expertise would be extracted from them and used to build the South Carolina low-country economy. The Esan of central Nigeria, the Akan and Brong of Ghana, the Mende of Sierra Leone, the Congo of what is now the Democratic Republic of Congo and Angola, a kingdom with its own complex political history and diplomatic relationships that was systematically dismantled by the Portuguese and their trading partners over 3 centuries. Each of these groups carries a genetically distinct profile, different haplogroup frequencies, different disease resistance variants, different HLA, immune gene distributions, shaped by thousands of years of distinct environmental and demographic history. The genetic distance between a Yoruba person from Lagos and a Mandinka person from Banjul is, in many cases, greater than the genetic distance between a Norwegian and a Greek. West Africa is not a monolith, it is a mosaic, and that mosaic is what the slave trade broke apart, deliberately, systematically, over 350 years. This is the baseline. This is what existed before the first slave ship left the West African coast. Now, the first thing the comparative data revealed, And this is where the surprise began, was just how specifically the West African ancestry in Black American genomes maps to particular ethnic groups. The landmark study establishing this was published in the Proceedings of the National Academy of Sciences in 2010, led by Carlos Bustamante at Cornell University and Sarah Tishkoff at the University of Pennsylvania. They analyzed genome-wide data from 365 African Americans, 203 West Africans from 12 different countries, and 400 Europeans from 42 countries. The West African component of Black American DNA, which averages roughly 73 to 77% of the total genome, was not a generic, undifferentiated West African signal. It was specific. It clustered most strongly with non-Bantu Niger Kordofanian speaking populations, specifically the Igbo and Yoruba of Nigeria and the Brong of Ghana. The Bustamante-Tishkoff study was the first to establish this precision at genome-wide scale, but subsequent research deepened the picture considerably. The 2020 study by Stephen Micheletti and colleagues at 23andMe, published in the American Journal of Human Genetics, analyzed over 50,000 consenting customers on both sides of the Atlantic and cross-referenced their genetic profiles against the Transatlantic Slave Trade Database, the most comprehensive shipping record of the enslaved African trade ever assembled. When they broke down the West African ancestry and Black American genomes by specific ethno-linguistic group, the Nigerian signal dominated. Approximately 35% of the average Black American's African ancestry traces to Nigeria, specifically to Yoruba, Igbo, and Esan populations. Ghanaian, Liberian, and Sierra Leonean ancestry contributed another significant component. Congolese and Angolan ancestry from the Congo and related Bantu groups formed the Central African layer. When 23andMe expanded its African ethno-linguistic matching in 2022, using 25 new reference populations from sub-Saharan Africa, the most common matches for African American customers were the Igbo and Congo and Mbundu people, each matching approximately 30% of African American customers. The Fula and Wolof of Senegambia matched approximately 13%. What this means is that when scientists compare Black American DNA to West African DNA, they are not comparing a diaspora to a continent. They are comparing a diaspora to specific ethnic communities, communities whose descendants are alive today, whose cultures continue, whose genetic profiles can be read with extraordinary precision. The slave trade did not erase those connections. It tried to. It failed. The genome remembers exactly who was taken and from where. Before we go further, if you are new to this channel, subscribe right now. We cover the genetic connections that history buried and science has recovered. This one is personal for tens of millions of people. Subscribe. Let's keep going. But here is where the data got genuinely surprising, because the comparison between Black American DNA and West African DNA also reveals something that nobody who thought they already knew this story had anticipated. The X chromosome told a different story from the rest of the genome. In the landmark Bustamante-Tishkoff study, and confirmed in subsequent research, the X chromosomes of African Americans showed elevated levels of African ancestry compared to the autosomal genome, the non-sex chromosomes that make up the bulk of DNA. This asymmetry is a biological record of the sex-biased pattern of gene flow during slavery. African women contributed more genetic material to the African American gene pool than African men, consistently, across generations, across regions. This pattern is consistent with the documented history of African women being subjected to sexual exploitation by European men with power over their lives, and consistent with the higher mortality rates among enslaved African men who were more often worked to death in field labor, exposed to greater physical danger, and more likely to die before leaving genetic descendants. The Micheletti study quantified this asymmetry in a way that made the historical record visible in the genome. Across the Americas, there was a very strong bias toward African women contributing to the present-day gene pool compared to African men. In the United States, That bias reflects both systematic sexual exploitation and the documented practice among slaveholders of deliberately managing reproduction within enslaved communities, pairing enslaved men and women to produce children who would expand the labor force without requiring new purchases from across the Atlantic. Joanna Mountain of 23andMe, who co-led the study alongside Micheletti, noted that the female bias is particularly striking given that the majority of enslaved individuals shipped to North America were male. The genome recorded the discrepancy. These patterns mean that when you compare Black American DNA to modern West African DNA, the maternal lines, the mitochondrial DNA, passed strictly from mother to child, show stronger and more direct African connections than the paternal lines. More than half of African American mitochondrial lineages trace specifically to West Africa. The paternal Y chromosome lines tell a different story, a European story, concentrated in the British and Irish genetic cluster, the biological record of who the fathers were and how those paternal connections were formed. Approximately 35% of Black American men carry a European Y chromosome. In the West African populations their ancestors came from, that number is near zero. That difference alone tells you everything you need to know about what happened between 1619 and 1865. Now let's talk about the most scientifically significant finding in the comparison, and the one that surprised researchers most. Not the similarities between Black American and West African DNA, the differences. Over 400 years, approximately 16 to 20 generations, the Black American genome has been shaped by forces that West African genomes have not experienced. The Middle Passage, the disease environments of the American South, forced reproduction, extreme mortality events on specific plantation types, the mixing of Africans from dozens of distinct ethnic groups who would never have interbred in West Africa, producing new genetic combinations that had never existed before, and 4 centuries of living in a completely different environment from the one that shaped West African immune genes. A 2023 Frontiers in Genetics paper led by researchers focused on what the team called legacy African Americans, the 40+ million multi-generational Black Americans whose families have been in the United States for multiple generations, noted explicitly that 400 years of exposure to American slavery, Jim Crow, segregation, and disparate health environments has uniquely shaped both the genomes and epigenomes of Black Americans in ways that make them a genetically distinct population from their West African source groups. One of the clearest genetic signals of this difference involves immune function. West African populations carry extraordinarily rich and diverse HLA gene profiles, the human leukocyte antigen system, the part of the genome that governs immune response and disease resistance. Research published in PNAS by Tishkoff's group found that the HLA diversity in West African populations is significantly greater than the HLA diversity in African American populations. 4 centuries of population bottlenecks, the selective mortality of the Middle Passage, the catastrophic death rates on specific plantation types, the near extinction of entire genetic subgroups like the Senegambians, who died on the rice plantations of coastal South Carolina, reduced the breadth of immune gene diversity in the Black American genome compared to what existed in the source populations. The Senegambian signal is perhaps the most striking specific example. The Mandinka and Wolof people of Senegambia were among the most heavily transported ethnic groups to North America in the earliest waves of the slave trade, specifically because they were expert rice cultivators. The shipping records are explicit. They arrived in documented numbers, and then they largely disappeared from the Black American genome. Not because they weren't there, but because the rice plantations of the South Carolina and Georgia Lowcountry, riddled with malaria and brutal mortality, killed them before they could leave descendants in sufficient numbers to maintain their genetic signature. A whole people's genetic contribution to Black America was nearly erased, not by time, but by the specific conditions of their exploitation. That erasure is visible in the data. You can see the gap where a people should be. The APOL1 gene tells another critical part of this story, and it is the part that has the most immediate consequences for living people today. Two variants of the APOL1 gene, known as G1 and G2, are found at elevated frequencies in West African populations and by inheritance in Black Americans. These variants confer significant protection against trypanosomiasis, the parasitic disease caused by the Trypanosoma brucei parasite and transmitted by the tsetse fly, commonly known as sleeping sickness, which has been a deadly and persistent feature of the West African disease environment for tens of thousands of years. Natural selection drove these protective variants to high frequency in West African populations across millennia, because carriers survived sleeping sickness at higher rates, reached reproductive age, and passed the protective alleles to their children. This is evolution working exactly as Darwin described it, selecting for survival in a specific environment over many generations. But here is where the transplantation from West Africa to North America created a genetic mismatch with consequences that are still unfolding in clinical settings today. The APOL1G1 and G2 variants, while powerfully protective against trypanosomiasis in West Africa, are associated with significantly elevated risk of chronic kidney disease, focal segmental glomerulosclerosis, and end-stage renal disease in populations no longer exposed to trypanosomiasis. Research led by Martin Pollack at Harvard Medical School and colleagues at multiple institutions established that Black Americans who carry two copies of APOL1 risk variants, either G1/G1, G2/G2, or G1/G2, have a dramatically elevated risk of kidney disease compared to both European Americans and West Africans who carry the same variants but live in environments where the trypanosome is still present. The numbers are stark. Black Americans with two APOL1 risk alleles have approximately 7 to 10 times the risk of focal segmental glomerulosclerosis, a severe form of kidney disease, compared to those without the risk variants. They have a significantly elevated risk of HIV-associated nephropathy, lupus, nephritis, and hypertension-associated kidney failure. These APOL1 risk variants are now recognized as one of the primary genetic contributors to the disproportionate burden of kidney disease in the Black American population, a disparity that has been documented for decades, but whose genetic basis was only fully established in the 2000s. This is what 400 years of living in a different disease environment does to an inherited genome. The West African adaptation that kept your ancestors alive in Yoruba land or along the Gambia River is now, in the absence of the pathogen it evolved to fight, contributing to a measurable disease burden. The genome carried the protective variant across the Atlantic. The pathogen it protected against did not come with it, and that mismatch between a West African immune adaptation and a North American disease environment is now playing out in doctors' offices and dialysis centers across the United States, encoded in a gene that was doing exactly what natural selection designed it to do. So here's what the full comparison actually shows. Black American DNA and West African DNA share a deep and specific connection, deeper and more specific than most people know, and more precisely traceable than most people realize. The dominant West African ethnic groups in Black American ancestry are identifiable, primarily Nigerian, clustered around the Yoruba, Igbo, and Esan, secondarily Ghanaian and Sierra Leonean and Congolese, with a traceable Senegambian thread that is measurably smaller than it should be for reasons the genome itself records with devastating specificity. The mitochondrial lines trace directly back to specific ethnic communities in ways that are increasingly precise as reference databases expand. The X chromosome tells the story of which sex maintained the African genetic connection most consistently across generations of violence, coercion, and the deliberate management of enslaved reproduction. But the comparison also shows something else, something that 23 and me and Sarah Tishkoff and the Catoctin Furnace research team and the Frontiers in Genetics Legacy African American Study all point toward, each in their own way, from their own angle of scientific inquiry. Black Americans are not simply West Africans who were relocated to a different continent and left to carry on as before. They are a genetically distinct population forged by 400 years of catastrophic selection pressure, forced admixture between ethnic groups that would never have combined in West Africa, reduced HLA immune gene diversity relative to source populations, APOL1 variants now operating in a pathogen environment they were never designed for, and the accumulated biological legacy of a disease environment their immune systems had 400 years to encounter but no evolutionary history to prepare for. They carry ancient West African lineages at their core, specifically, precisely, traceably, in ways that are now measurable at the individual level by any consumer genetics company with a large enough African reference panel. And they carry, in the specific shape of those lineages, and in the specific ways those lineages were altered by the conditions of the Middle Passage and the plantation, the most detailed genetic record of a forced migration in the history of DNA science. Every percentage point, every missing haplogroup, every APOL1 allele, all of it is history, written in biology. The genome is both archive and biography. It tells you where you came from with a precision that slavery's architects could not have imagined would ever be readable. And it tells you what happened after you arrived, in the immune variants, the reduced diversity, the missing Senegambian signal, the European Y chromosomes, the APOL1 mismatch, all of it encoded in 4 letters repeated across 3 billion base pairs. The comparison surprised scientists because the data was more precise, more layered, and more historically specific than any prior model had predicted. It was not simply the story of a diaspora carrying its homeland's DNA across an ocean and arriving unchanged on the other side. It was the story of what a genome looks like after it has survived something specifically designed to destroy it and survived anyway, carrying the original signal intact beneath the damage, altered in specific and traceable ways, and readable in extraordinary detail by anyone willing to look closely and honestly enough at what the data says. The bones don't lie, the genome doesn't forget, and the West African DNA that forms the foundation of Black American ancestry has been waiting, patiently, precisely, across 400 years and 16 generations in every systematic attempt to erase the record, for the science to finally develop the tools to read what it has always held. It held everything. The science reads it.